Thank you very much. And I'm very upset because you always said I was your first customer. Now Kieran is.
He was the first consulting customer. You were my first customer.
Anyway, so we're Revolver Therapeutics. Nothing to do with the guns, unfortunately. It's actually to do with the shot with the proteins, which we'll come on to. We have developing peptides to translate transcription factors into therapy. So we're shutting down transcription factors to treat cancers. You have the next slide, please. So transcription factors that many of you know are proteins. They bind to genes and they turn them on and off. So they're like sort of switches for genes. Aberrant gene expression is driven by these, and they're seen in many cancers as master regulators of disease, triggering many aberrant gene expression. But they can be blocked to treat those cancers, or that's our hypothesis, but also they have relevance in other diseases as well. A big number here, big number warning, but we basically looked at this on the basis of upfront payments for early stage deals in transcription factor areas. So it's not all BioWorld dollars, it does include some real cash as well. But I think, if we look at some of the competitors, for example, Curve on the bottom, they've got very substantial upfronts, and they're not a big Californian biotech, they're down in Southampton. So basically what we've developed is peptides to inhibit what are historically thought as undruggable transcription factors. Good targets, very difficult to intervene therapeutically and to penetrate this new market. And we think there's early revenue streams. Obviously, we don't have any revenues yet because we just had our first investment, but the plan is to do deals with, significant upfronts. So if we move to the next slide, please. So this is a cartoon of the transcription factors showing the helical proteins here. And what they do is this end here binds to the DNA. And if we can disrupt that, we can sort of shut them down. And basically, these things that were seen as undruggable can now be modulated with the peptides that we are discovering with reasonable affinities. And beyond the ones we're currently focused on, there's 1,600 different transcription factors. We're only focusing on a relatively small number that are relevant to cancers. Next slide, please. So, this is the Goldilocks solution, as they say. We're not too small and we're not too big. So we think by going for what we call Revolver peptides of around 2 to 6 kilodaltons, we can actually still get good penetration into cells, and we'll explain how. We can get around the high clearance problems seen by some of the smaller peptides, but also, more importantly, we can actually interact with the protein-protein interactions that we have to disrupt to make this work. Basically, small molecules are a little bit too small. And so we think we're just right for doing the task that we're asking these molecules to prove. Next slide, please. So this is the technical solution, or the series of technical platforms that have integrated in Jodie's lab, and I'll introduce him in a bit. We've broken them up because they're protected by different patent estates. But basically the core technology, the core platform is transcription block survival, or TBS, where sort of libraries of 50 million or so peptides, so covering a fairly large chemical diversity space, can, unlike a sort of phage display type assay where you're just looking at binding, is looking at the ones that just shut down the process. So we're selecting out of the 50 million in each library the ones that are functionally active and not getting distracted by the ones that just bind and don't do anything. The other thing is the cross-linking penetration. Obviously, we're going for transcription factors. That only works if you get them into the cell and into the nucleus. So we need to get them into the right place at the right time. We have an informatics platform. We're not using the word AI because I think it's been, everyone's using it at the moment. But yes, can we do machine learning? Yes. And then we can also attach cargoes as well, not in the sort of classical sense, but in a way to enhance the function of the peptides that we're developing. So that's the platform. We can ask more questions later as we go through. So next slide, please. So why invest now? This is probably the most important thing. So, you don't want to be too early, you don't want to be too late. We think obviously there is an urgent unmet medical need in cancer and elsewhere. We think transcription factors have been seen as the holy grail, but sort of not druggable, and we think we've solved that problem. And what we've done, we have an integrated functional cellular and in silico technology to make that work. Currently, we're targeting cancers, initially pediatric high-grade gliomas with the Institute of Cancer Research funded by Innovate UK. Acute myeloid leukemia, colorectal cancer and prostate cancer, largely because the transcription factors that we're modulating are relevant to these diseases. And we believe they can be optimized from being early stage peptides into drug-like molecules. And we can talk a little bit about that later if you're interested. So next slide, please. So who else is out there? This is just a list, and the surprise is they're not all in the US, they're not all in Boston and California, some of them here in the UK. And I mentioned Curve Therapeutics before. They've actually done a major upfront deal with MSD with, real cash upfront, not just all BioWorld phantom dollars. So actually people working in this space can actually do big upfront deals on multi-target deals. So where do we fit in? We believe we're the only ones who can screen the helical interface between the transcription factor and the DNA itself, where it actually binds, with a functional cell-based readout. So we're, going through, multiple iterations. If you're doing it with small molecule design, make, test chemistry, it would take you forever. We can get there a lot faster by using the power of evolution to get there. So basically what the platform does, it enforces the entire peptide libraries into conformations that cannot normally be accessed by, conventional, let's sort of see if it binds to a phage display type thing. And we can select for the peptides that ensure the target protein binding and complete shutdown. So we've saved a lot of the design, make, test issues that you often get in traditional drug discovery programs. So if we go to the next slide, please. What's our business model? It's a hybrid business model, which means we'll take cash, but we'll, continue to do our own R&D. And really looking at sort of platform partnering along the lines of Curve and others, where you do multi-target upfront deals for cash, that enable you to raise money when you need to raise money and not raise money when you have to. And basically continue to push our assets forward as far as we can for sort of out-licensing deals, the sort of classical, biotech model that we were talking about earlier. So we think we have a rapid road to revenues. We don't have any yet, so we're probably a little bit early for someone like Kieran to invest, but we hope to be revenue generating within years, not decades. Can we have another slide, please? So my background is originally I was an academic, and not quite in the time of the Doomsday Book, but around the time probably that this bakery was built. Was a therapeutic area head at large pharma for a while, and been through now multiple exits from early stage. So half of those were roughly around licensing transactions, but also half of them were in CROs and research tools. And actually the last one was to Danaher, which was the company that Kieran was talking about earlier. Jodie, who you can meet later, and we haven't got pictures here because we've got the best looking team here at the moment today, is Jodie's the Chief Scientific Officer, 25 years. He actually started the technology, patented the technology out of the University of Bath. He and I worked in a company that I now learn is the third, customer of O2H or the old form. So we worked together in the peptide space before working with O2H. And, he's been really driving the team from the University of Bath. Jenny's on the board. She's actually tech transfer for the University of Bath, but also works for the Sanger down the road here. Keith, I think, is known to many of you through multiple interactions and is joining the board as a non-exec, and he was formerly CEO of CRT and Storm Therapeutics. And Andrew is really the guy who's been driving the technology at Bath and is, we call him the Director of Platform Technology. Our advisory board is David Fairley in Australia, Randy Mesney in Bath, and Chris Jones, who's our collaborator at the ICR in pediatric glioma. Have the next slide, please. How long have I got? 2 minutes. So what do we want to do? We need about £500,000, really to extend the 150,000 that we've raised from O2H and Future Planet Capital, who are a bioengineering fund. They have another 35 million to deploy in the UK, and also match our grant, non-dilutive grant of just over £500,000 from Innovate UK. So what do we want to use the money for? To screen further targets with patented Revolver assay platforms, so not just on childhood cancers, but adult cancers as well. Optimize the active peptides for potency and delivery. We can get them into the cells, but are they staying there long enough? Is the residency time good enough? Is clearance too high or too low? We need to answer those questions. And complete, 3D organoid type studies and in vivo studies with our partners. And we're accessing those sort of models at the Institute of Cancer Research. And then file composition of matter patents to protect what we discover, because at the moment we've got platform patents that obviously protect what we do, but we want to very selectively file composition of matter around the things that we think might be drugs. And then the plan is to validate the platform and then do partnering deals, as we've just discussed. So I think that's it, and I finished 7 minutes over time, so do I get mad? But questions at me?
Given that you mentioned phage display, I take it you're not using a display platform, certainly not a cell-based platform. So by that account, can you incorporate non-naturals into your large peptide library?
What have you sort of non-natural peptides? or
Non-natural amino acids.
Yes, we can, can't we, Jodie? I think we can.
We haven't done it yet, so we can incorporate non-natural. Some of the peptides are sort of non-natural overall by virtue of the constraints that modify them during screening.
Yes.
So we haven't specifically put in non-natural side.
Incorporating non-natural amino acids into peptide libraries has been a holy grail for quite some time. So we really thought we can do it.
I mean, it can be done, can it not, through anti-suppressor and the heroic series stuff is.
Yeah, but the sort of the tRNA approach is it's been around donkey's years since I first started in the peptide field, and it's not great. If you can do it synthetically, it's going to be a lot more reliable.
Well, we can synthesize, but obviously not at the rate that we can in the TBS platform. So clearly, you know, we do synthesize and test,
but it's not in your naive library.
It's not currently in the naive libraries.
Yeah.
But the post-translation modifier.
It's a similar question. I mean, look at, I know David very, very well, and he's done well. And then Max Smythe, you probably know, who did system peptides, which became Protagonist, which now has the best-in-class R23 drug on the market with J&J. So, long answer to that question. In your compound progression plan, are you putting enough filters early on ADME, bioavailability, biodistribution? Because in the peptide space, you can get great potency, cell-based activity, yada, 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 but then you have a lot of ADME distribution issues. So
yeah,
so are the filters coming on early enough to get a drug?
Well, obviously we're testing those. There's sort of certain things we can do in Bath. You know, we don't have in vivo facilities at Bath, but we can access that through CRO. So we've been working with, in fact, our first grant from Innovate UK funded that work at Axis Biosystems in Northern Ireland. The other person we're working with is David, obviously, and he has access to a lot of those materials in Queensland. And the ICR have a very sophisticated panel of triage models. Obviously, we can only access those for that specific grant. But yes, we're working with partners who have access to the appropriate, in vitro and in vivo model. And then the idea is that we will take those data and then use it to help optimize and model the patterns. So the big thing is, why does drug discovery take so long? You have to go through multiple iterations. So the idea is if we can use things like decision theory to reduce the cycles of design, make, test, then we can get faster. Have we done it? No. Do we have access to the assays that can give us the data to make decisions? Yes.
So thank you very much.